A very good evening to all. Today's medical term is once again something different because we are going to discuss about a cell, a blood cell, a white blood cell, T lymphocyte. T stands for the thymus. But why thymus? Because as we know, all blood cells are born, develop, and mature in the bone marrow from the hematopoietic stem cells. So, what is the role of thymus gland in blood cell production? Yes, as I mentioned before, all blood cells except the T cells, they are born in the bone marrow, but they develop, they mature in the thymus gland. So, what happens is, as the bone marrow produces these immature T cells, or the T cell precursor, it travels to the blood, reaches the thymus gland. And that's where the development process known as thymopoiesis is initiated. So, this process occurs in three stages, namely double negative, double positive, and single positive. So, what happens in each of these stages? Well, at the double negative, the thymocytes are at the earlier stages, so there is no differentiation whether is it a T helper or a T cytotoxic cell. So the TCR, that is the T cell receptor genes, are rearranged, and there is a beta selection process that happens where cells with the functional TCR beta only is allowed to be surviving. Then comes the double positive stage where the T cells starts expressing the CD4 and CD8. And at this stage, there is TCR alpha chains get rearranged, and this is where the MHC restriction happens. And in the single positive stage, that is where CD8 plus, which is able to be recognize MHC1 survives, and CD4 plus, which can recognize MHC2 survives. So the double positive and double negative stages are two critical processes. So only the T cells which can bind to the self MHC will survive, and those who can't undergo apoptosis, known as positive selection. Whereas the ones who can bind strongly to self antigens are eliminated, and this is known as negative selection. So, as the maturation occurs, both rounds would exist and T cell selection do take place.